Anouk, thank you for joining us today to share the beautiful memory of Lhasa. Thank you to all of our families for your impactful quotes, for sharing your memories and your images in that video. And thank you to the Boreal Collective for creating the video. To all of those with us who have lost a child, a family member, a friend to KAND, we hold you in our hearts. We love you all so very much. And we are so thankful for your presence here today. Next, we are going to move into a conversation that I had with Hans Christinger, KFNA.org board member, and Kat last week about our therapeutic development strategy.
Right. Welcome, everyone, to our conversation on KFNA.org's therapeutic development strategy. I'm Kathryn Atchley, president of KFNA.org and mom to Parker. And with me, we have Dr. Dominique Lassard, chief science officer of KFNA.org, and Hans Christinger, board member of KFNA.org. And I think the conversation we're going to have is really important for everyone in the community because since KFNA.org was founded in 2017, we have been really focused on not only providing support and advocacy for our families to improve the lives of patients, but also driving research and therapeutic development. And we know this is really important because every day that we go without treatment, we know more and more children and adults are going to lose their mobility, lose their sight, their speech, even maybe their ability to eat or breathe, and even their lives. And so this is why our mission is really so urgent, that we're so focused on changing this fate of KAND by rapidly discovering treatments and cures. And, you know, when I think about all the progress we've made over the last few years, I credit every single person in this community for that success, and that definitely includes both Dr. Dom and Hans. So thank you both for this conversation and your leadership. So to get started, why don't we have Dr. Dom introduce yourself and tell us how you got involved in the KFNA community.
Sure. Hi, everyone. I'm Dr. Dominique Lassard. To put a face to a location, because we're all sort of scattered around the world, I currently live in the state of Vermont in the United States. And I first became involved with KFNA.org as a volunteer while I was finishing up my PhD at the University of Vermont. So my PhD was focused on KFNA, specifically understanding the KFNA protein. And one day when I had a publication of mine making the rounds on Twitter, I ended up connecting with Luke and Kat over Twitter, and we started chatting about ways that I could get involved with the organization. And what this amounted to ultimately was a series of projects focused on helping uncomplicate, demystify some of the language that's used in scientific writing about KFNA that can be a huge barrier to our understanding of what's actually going on with our own bodies, with our family members' bodies. That ultimately led me to going to the 2019 in-person KAND conference where I gave a presentation. And I always like to talk about that experience as truly life-changing because it was. I left that conference a really different person and carrying a very different perspective about what it means to be a scientist, solely based on the interactions that I had with the KFNA community and everybody's families and getting to see everybody together. And because of that, I pretty much changed my career trajectory. And once I was able to complete my PhD, I hopped on board as a member of the leadership team at KFNA.org, thanks to the CZI RARES 1 grant that we received. And I've been with the org now for about a year and a half.
Thank you. We're so glad you're here. Very lucky to have you. And Hans, what about you? Introduce yourself and tell us how you got involved with the KFNA community.
First of all, well, I'm Hans Christinger, and I'm a board member of KFNA.org. And Kat, thanks so much for having me. Nice to be here with everybody. It's really an honor to be here with you guys and with everybody in the community. So I got involved with the organization really through Luke Rosen, my good friend Luke. We got to know each other at the company where we worked together at Therapeutics. Luke had just joined us as the head of patient advocacy. My day job there, I'm vice president of business development and alliance management for Therapeutics. And Luke and I hit it off right away. Similar mindset. A passion for driving change and doing things quickly and efficiently, and just really like-minded. And one day Luke approached me and asked really kind of politely, I hate to ask you, but do you know somebody in this particular company, because they had some interesting science that was applicable for KIF1A. And I said, of course, so I connected him with people that I knew, but then said, Luke, I've been doing this for a long time for a number of different companies, big and small. Let's sit down and go through a little bit more of a strategic approach. We'll continue with these one-offs, but maybe we can put a broader strategy together. And that kind of kicked things off. Since then, we've worked closely at Ovid and with Wendy Chung and the team at Columbia to put a collaboration in place where KIF1A is really the lead, the example, the flagship program, if you will. And we've done a number of things, and that has broadened as well. And we'll talk a little bit about the research network. So it's been really fantastic working with everybody here and trying to get things set up and do things efficiently and quickly. So that's kind of how I got involved with the org. Should I go in a little bit about the background, or is that good enough for the first answer?
Go. I'd love to hear a little bit about the background. Yeah. Go ahead.
So I've been doing this for a while. It's been about 30 years overall in the biotech and pharma world. The last 7 years here in the New York, New Jersey area have been working with kind of small companies, building leading companies focused on science-driven, patient-centered organizations. Ovid Therapeutics is focused on really transforming medicine and care for those living with rare neurological disorders. And we seek out the best science, we seek out the best teams, and find ways where we can bring our strengths and have an impact in those areas. So these are things like Angelman syndrome, KIF1A or KAND, rare seizures, things like that. Prior to that, I was doing the same thing, trying to tackle the leading causes of blindness at a leading ophthalmology company, also in New York. But I came with a large amount of time in large global companies. So companies like Roche and Abbott, we moved here from Switzerland. You look at Roche, well, at that time, I don't know what they have today, but at that time they had a $10 billion research and development budget. And while they had a massive impact, or they are having a massive impact in areas like oncology and in the areas that they're working in, they recognized that was an insufficient budget. That was small compared to what the rest of the world was doing. So a very targeted and specific approach to comb the planet and understand where the best science is and where the best teams are. And again, figure out what could we do as Roche to partner with those teams to really accelerate the good science that was being done there. What could Roche bring to the table to drive change and have impact for patients and families globally? Same story with Abbott. Same kind of concept. So I've seen it from the big company perspective. I've seen it from the small company perspective, and now the foundation perspective. And just to go back to the early days, I spent the 90s at Genentech, actually in the lab, looking at and understanding important proteins in human biology, and trying to figure out what can we learn about them so that we can create medicines. And the example for my project was something called vascular endothelial growth factor, or VEGF, which they've turned into quite important medicines in oncology and also in treating blindness as well. So 2 programs that came out of that. So that's a bit about my background.
Yeah. So how, I guess in a nutshell, how do you apply all that experience then to help with KIF1A.org's therapeutic strategy?
Yeah, so I think, there are a couple of things here. In all of those, I've made a lot of friends and a lot of contacts around the world, scientifically, medically, and so bringing those contacts and relationships, and I'll say I'm never the smartest person in the conversation, so I can bring the contacts and relationships with people who are smarter than I am, but also just an understanding of strategy and tactics as well, right? So I've seen a lot of things that work well. I've seen a lot of things that don't work well. I see a lot of things that waste time and things that we can do better. So it's kind of seeing the puzzle pieces and being able to work together to bring the right people to a conversation to figure out how do you put those puzzle pieces together. And just to illustrate it, last year there was about $12 billion poured into neuroscience. And that's not counting the research and development dollars that the companies are putting in. That's just looking at the amount of money that investors poured into, the amount of money companies raised in order to put into R&D for neuroscience, and the upfront dollars that went into partnering agreements between companies in neuroscience. So it's a good proxy, and it's a big number, $12 billion, right, in 1 year for neuroscience. And what can we do at KAND or KIF1A? Well, we've got some great models. You've got some mouse models, some cell lines that are derived from patients and mutations and different models that we have. So we can create a platform where when we see good science, we can apply that science to this filter, and you can filter that very efficiently and inexpensively. You can quickly filter that $12 billion of expense that other people are putting into all kinds of other disorders. Great amount of research, great amount of effort, not enough effort on KIF1A. So we can bring our very simple tools and platform and just filter their money into our area and try to do the best thing to accelerate science and medicine for KIF1A.
Exactly. And that a lot of that makes me think of Dom and your day-to-day work with the organization. So I guess as Chief Science Officer of KIF1A.org, what does your day look like?
Honestly, my days look a little bit hectic, but in a really rewarding way. We have a lot of moving parts in the back end of the organization. And for me, when I think about, I'm starting my day, turning on my computer, getting ready to work, the mindset and the motivation that I carry with myself every day, which infiltrates in everything that I do, is we need a treatment, we need a cure, we need to work as quickly as possible, as efficiently as possible, and we need to importantly inject the community voice and the patient voice into every single thing that we do. And so a couple of things that I do throughout the day, I do a lot of company correspondence. So one of my roles as CSO is to educate, to provide resources, to pitch projects to a whole slew of companies. If I were to pan my camera to the right, I've got a whiteboard over here with about 30 different companies that we're in various levels of conversation with. So we're talking to a lot of people at a wide range of levels. There's also an extensive amount of management that I do of the research network. So our goal in having a research network is we want to support our research network members. We want to support the people that are physically doing the work in a variety of different ways to help us find a treatment and a cure. And so this is kind of project management, but also acting as a resource, as an organization, to speed up that research process in any way that we can. And right now that's at about 100 members, our research network.
Wow.
So it's grown quite a bit in the past year or so. It's
year and a half. Yeah. So, I know that we're grateful for both of you for, your expertise and your guidance as we're figuring out how to drive research and therapeutic development forward. But we're also grateful for this growing research network that also plays a big role. it's a great network that we can also lean on for guidance on strategy and how to move forward. So can you talk a little bit more about that research network and, you know, who's in it and what role do they play?
Yeah, I'll give a quick visual example of just some of the people that we have in our research network, because as I mentioned, it's growing, it's quite large. But I like the slide because it shows how interactive and hub and spoke, as we refer to it, a lot of these different therapeutic approaches we have ongoing, and how our research network plays a role in making this all possible. So the research network is really our formalized entity of scientific stakeholders, and collectively they advance and accelerate therapeutic development for KAND. This contains academicians, biotech industry, government partners, other organizations that play the important role of the ones that are physically doing the work, like I mentioned before, because we as an organization, we can facilitate and we can manage and aid as much as we possibly can, but we're not in labs, we're not seeing patients, we're not having these internal conversations within these high level companies. And this is where we rely on the research network. But by being able to formalize this entity is a lot of work and requires a huge increase in capacity on KIF1A.org's end, which our CZEI Rare as One grant has been instrumental in this process to increase the capacity we need so we can provide resources to our research network to make them successful.
Right, absolutely. And so kind of shifting gears a little bit, you know, again, kind of reminding ourselves why we're building this research network and working so relentlessly. I think the number one question on a lot of minds of families is, when exactly are we going to get to the first clinical trial for KAND? You know, because again, we know every day that goes by without treatment, our relentless superheroes are, you know, declining and facing an uphill battle. So how would you answer that question, both Dom and Hans? When do you think we'll get to the first clinical trial?
I'll go first. Yeah.
Yeah.
I mean, it's a tricky question to set a timeline for, and that's because we are at the mercy of the pace of science and investigation driving therapeutic development. And, you know, as an organization, we move very quickly to get to clinical trial as quickly as possible. But as I mentioned in the video that preceded this session, that there are two components to everything that we do that drive us to the clinic, which are risk and failure. And failure feels terrible, right? It can feel futile and it can feel upsetting, but it's important for us to remember how important failure is in the process of scientific investigation. And so for us, it's not about trying to prevent failure, because we know it, to a certain extent it's going to happen, but it's about making sure that the failures that do happen are addressable and don't cause, you know, an irreversible level of harm to our mission. And the same thing goes with risk. Risk is this constant assessment between risk and reward on our end, because each type of project we have is going to have a certain element of risk, because nothing is guaranteed to work. But I think that there's a big difference between having a well-calculated risk and having a poorly thought out risk. And so on our end, we make sure that the risks we take are as vetted as possible, as planned out as possible. And we're pulling in all the right people that we need to help give us their information, their opinion, pulling in from our research network to get their understanding so we can make the most vetted risks possible when we're advancing our therapeutic development towards clinical trial.
Yeah, and I would add to that, you know, we recognize that no matter how much good work we do, there's always a balance between urgency and panic. And we know that every day. And every day we wake up, our mission is to try to move things as quickly as possible to get to the clinic. So, you know, what are the kind of things that we think about and how do we approach that? I'll make a couple points on that. And then second, a little bit of what does it mean exactly. So we try to make sure there are several things tried at once, right? We don't do things sequentially, we do things in parallel as much as possible. We look to see where we can borrow things or take things from other areas that will accelerate our learnings or our different approaches. And we're always looking to front load things. If you find an opportunity that looks like it's going to be successful, we say, okay, can we accelerate certain things and front load those activities where traditionally in drug development you might wait for more data. Let's not wait for data, let's do it in parallel. So, maybe just to give a little bit more clarity, what does all of that mean? Doing things in parallel, front loading stuff, and borrowing things from other places. What does that mean? So first, to create a medicine, there are certain things that you need to show to the FDA. But more importantly, the parents and families want to have the same assurances and the same comfort. It's not just the FDA, it's for us and it's for the community that we need to show these things. We need to show that the medicine that the program that we're taking forward as a medicine is safe. It's not going to cause any harm. That's the first thing. The second thing is you want to show that the scientific hypothesis makes sense. There's a rationale there. And so those are the two things that you set out to do. And how do you do that? There are a standard set of, not standard, but there are a set of things that you try in preclinical testing. So in an environment before you get into testing these in humans in clinical trials. And they include a bunch of different things, animal models and things like that. And then there are a number of things when you get to the clinic that there are a number of kind of tried and true programs in clinical testing that you try. So how do we accelerate these things? There are a couple of things where, because we understand the gene, because we understand the mutations in the gene, we can accelerate research by directly targeting genetic effects. So these are things where if you show in a petri dish that it works, then you can forego certain animal models because it just doesn't make sense. The genes are different in animals. It's a smaller subset of animal models than you would normally try for other therapeutic areas that you're working in. So that way you can go a little bit faster and you expect to have a more robust signal because you're directly addressing the genetic deficit. Then when you get into the clinic, the FDA has recognized that working in small populations is different than working in large populations. So there are agreements, and these are things that we negotiate with, the community negotiate with the FDA in parallel all through the process to make sure that we're doing things right and we're not wasting time and energy. But when you get into the clinic, you can actually take a tip of the spear approach. You can go with a smaller set of patients, and because you're going after genetic deficit, you're probably seeing a bigger effect. So you can your clinical trials can be shorter and faster because you are using a smaller set of people. And then because of the rare pathway with the FDA, you actually can use a smaller number of clinical trials. So you don't need to do multiple clinical trials. You can actually have a more targeted set of trials. So it's all geared to being faster and more targeted and more specific in how you are creating medicines. So, those are some of the different tools and approaches. Again, if you think about it, we part of our portfolio approach is to make sure we're taking genetic approaches so we go quickly. We also, you may have heard around repurposing. So you can take libraries of medicines that have been approved by the FDA or have been in the clinic, and so there's a safety profile, and you can filter those through our models, whether it's the mice or whether it's the cell-based assays or cell-based models that we have, and you already know that there's a certain amount of safety, so you're comfortable taking those into the clinic. So that there are a number of different places and areas that we can try to be faster and more effective. So hopefully I'm starting to babble. Hopefully that gives a little bit of a picture of what we try to do and what it means to do things in parallel, what it means to try to borrow things from other places, and what it means to try to front load. Front loading can be, we have to show that medicines will not cause a problem. They need to have a certain amount of stability. They need to have a certain amount of safety over 6, 9, 12 months, right? So what can we do? Do we wait for the perfect data in order to test those things to show that in 6, 9, 12 months they're safe and effective? No, we can, when we have a good idea, maybe we can start those earlier. So we always look for those trigger points as well. But maybe I'll pause there and see if that answered the question.
No, I think that's really helpful. And just looking at this slide here, I'm thinking of our phrase we like to say, that we're taking multiple shots on goal. We are working in parallel. We're not trying one strategic approach, seeing if it, passes or fails, and then move on to the next. We're trying to do multiple things at once. So I guess with that in mind, what do you think is achievable for us, or what are we trying to achieve by the end of this year, by the end of 2021?
And maybe I can make one comment, if I can, Dom, before you jump in. But when you look at this, there are what does multiple shots on goal mean? I mean, I might have the wrong number, but I count about 8 different approaches. When you look at Ovid and the work they're doing, there are several approaches within what they're doing, and going after genetic-based therapies. Ionis is another one. I don't have all the details there, but that's another approach, very similar. We've got MCRI in Australia that is doing a lot. So, there's so many different things. And then to your question, the foundation is now setting up an iPSC platform to complement what's been done with the mice. And I know of about 5 different programs there that we can put through, borrowing from other concepts where you're looking at neuronal cell health and things like that. So it's a very powerful platform that I don't know if that completely jumps out. I mean, this looks really impressive, but the numbers behind it are even more impressive. And I personally hope that we can have the iPSC screening platform up and running by the end of this year, and have some compounds in it by early next year. But I don't know, Dominique, you probably have a better answer to that question.
I think I have a very similar answer. Going back to the idea of shots on goal, we have many plates spinning in the air right now, with the expectation that some may work and some may not. We hope they all work, but the reason that we spin so many is to have our multiple shots on goal. And I guess for a end of year goal, what I think would be feasible and fantastic would be to have a clear target that is at a stage in which we can start thinking beyond preclinical development. And as Hans mentioned, that doesn't have to be tied up in a box with a beautiful bow. It doesn't have to be perfect at that point, but having clarity beyond, okay, we have an understanding of how this compound, this target, this treatment, whatever it may be, because we have many options. We understand how it's working to a certain degree on a preclinical level. Let's enter the next year and start thinking about how we can make this transition into a clinical level. And by setting up these systems that Hans just mentioned, complementing mouse models with our iPSC lines and creating this high throughput industrialized system to get those answers and to get those translatable answers more efficiently and quickly are going to be huge and key to making that transition.
Right. Yeah. And I think, on Saturday, we'll hear from a lot of our collaborators, the progress that we have been making and with a lot of these shots on goals. Another thing that I think about a lot is, I know we talk a lot about de-risking the process for industry. So can you explain a little bit about what that really means and how that's helping us get ahead?
You want me to start, Dominic?
Go for it.
Yeah, I mean, that's a great question. As you look at this, everything we're doing here, we're making available for everybody in the research community. So this is really a long-term objective with really near-term impact as well. So what we hope to do with these screening platforms is that's available for, we identify Ovid, Ionis, MCRI, but there are other companies that we're speaking with. Dominic mentioned up to 30 or so. Hopefully this is a platform that generates those things that quickly can go to the clinic, but it's really enabling everybody else to, they all contribute to what we're trying to enable here. We share ideas on how we do things and make sure that what we're doing is appropriate and has the best input from the entire community, but then it is also available to everybody. So by doing that, hopefully we've got a number of shots on goal. We've lowered the time, money, and risk for companies to come in and bring their best magic sauce and move as quickly as possible. And and treat this as really almost, to use a buzzword, personalized medicine within KIF1A, right? It's you will have more than one treatment coming out of this. It's my expectation is not that you have one magic bullet, but you've got multiple cares and cures that are available through this.
Yeah. And I think when we talk about what is de-risking for industry partners, you have to understand what is risky for those partners and help ameliorate that risk. And so when we talk about de-risking, we recognize that guesswork and unknowns are risky in partnerships for industry partners. So we try to remove as many unknowns as possible. And we do so by having these tools for development that Hans just alluded to. These can be things like, data about our clinical understanding of KAND, our natural history study. That's huge. That's such a valuable resource in generating these relationships. Our mouse model is another tool for development. Our iPSC screening platform is going to be a fantastically large plus in generating these relationships with industry partners and trying to understand what would potentially be a barrier for a partnership to occur, understanding what barriers we can address, and then addressing those barriers before they even are at the table. So when we have these initial conversations with people, we can say, you don't have to generate a mouse model. You don't have to get patient-derived lines. You don't have to start a natural history study. We've done it. And we've done it, and we've facilitated our network members to do it in a way that it promotes this cross-collaborative sharing, which is also so valuable in forming these relationships.
Yeah, it amazes me, when I join these calls with you, Dom, talking with different partners and potential partners, biotechs that we can work with to develop treatment. The same questions always come up. Is there a natural history study? Is there a patient registry? Are there animal models? Is there a cell line, a model system we can use? And so we absolutely are, checking those boxes, and it just makes it so much easier for these collaborators to want to work with us. It's hard to say no when we've done so much up front. We're making it really easy for these partners to get involved. And another thing I've been thinking of is we've been talking about treatments and cures. So what is treatment versus what is cure? Is it the same? Are we using the same word to describe the same, or two different words to describe the same thing? Or are they different?
That's a great question. I think those terms are used interchangeably depending on who you're talking to. And so to give some perspective about when we're discussing within an organization, how we separate the two, When we're talking about a cure, we're likely talking about some sort of genetic editing therapy, basically something that could go in to someone's DNA, identify a specific KIF1A mutation, and correct it back to what it would look like if it was not mutated. And that sounds kind of like sci-fi, but what's remarkable is that these scientific tools do exist. But it's also important to note that these tools are still in very early stages of development. So while they may not necessarily be quote unquote ready for something like a KIF1A project, depending on the types of tools we're looking at, it's also important to note that we can still make progress towards these type of treatments by forming relationships with the companies and researchers that are currently developing this technology to get KIF1A on their radar. And that's exactly what we're aiming to do. We're working hard to make sure that when that technology is advanced enough, we are at the front of the line to get this type of therapy to our community. But in the interim, we can also talk about treatment. And when we say treatment, we're talking about the idea of alleviating certain KAND symptoms. And while that's not necessarily a cure, and while we're always striving for a cure, there's still so much positive impact that could be achieved with certain treatments. You know, it would be fantastic if we found a treatment that was able to slow the progression of certain KAND symptoms. And so when we talk about treatments and identifying candidates, it's also important to make sure that us as an organization are focused on meaningful treatment. What does a meaningful treatment mean to our community? And this is an example of why it's so important for us to hear community perspectives and what meaningful clinical outcomes would be for our community, because that type of perspective infiltrates everything that we do and infiltrates all of the relationships and technology and tools that we are seeking out and bringing on board to ultimately get to that treatment and to that cure.
Right. Yeah, absolutely. Another thing the community has been reflecting on as well is over the last year and a half, we've seen this incredible rapid response to COVID. And so, you know, I'm wondering, what do you think we can learn from that? And what do you think we can do differently? What have we been doing differently, and what can we continue to do differently to move faster at that COVID pace? Can we replicate that with KIF1A.org?
Yeah, and I think that's a great question. I think that's what we've been trying to do. I mean, a lot of what COVID brought was a tremendous awareness that really unified people to a particular mission and a particular cause, and that's what we've been trying to do. And what you saw was a tremendous number of shots on goal. You know, the amount of trials that were started, the amount of different approaches that were tried, and look, the first ones out were a totally novel kind of therapeutic approach with mRNA. So I would say there are a lot of similarities. The approach that worked there, I think, is fundamentally what we've been trying to do from day one here, which is, you know, just raise awareness, be out there, be vocal, make sure that all stakeholders in our ecosystem understand who we are and what we're trying to do. And that is at every aspect. And it's also raising awareness in the scientific community so people know to look for us. It's raising awareness in diagnosis so people know to look where they haven't looked before, and they put there's every aspect of this, there's awareness. Multiple shots on goal, we talked about that, right? Bringing all the world's resources and expertise into conversations to try to tackle this. That's what we've been doing, and we need to put our shoulder behind that every day. All of us have a role in that. Yeah.
What about with us all having a role to play in that? What about patients and families? What can we do to keep driving things forward faster than ever?
Yeah, I mean, I think of the butterfly quote, right? There's no small contribution. You have the butterfly in Asia that flaps its wings, and you have a weather change in New York and Mexico, right? It's true. You've got some things. There have been tremendous contributions and generosity throughout the community and the ecosystem, and some of them are easy to see. And I'd say everybody has a role to play, and there's nothing that's small, and it can be just from raising awareness, speak in the community, neighbors and schools and things like that. It's spending time with Wendy and the physicians and helping us understand what's meaningful, as Dominique said a minute ago, right? I mean, those types of things feed into how companies like Ovid think about clinical trial design and what's meaningful and how do you measure it. And those get rolled into FDA interactions. They also get rolled into what should we be looking at in these different preclinical models. There's so many things we could talk about that are important that we need to do, including fundraising. But it's really, everybody has a skill, everybody has a passion, and sometimes it's not always obvious. We don't think about what we're good at that we can bring to the table. So it's just being plugged into the community and sharing things, and you just never know. It can be telling the story simply for what you or you as a sibling are going through, right? That helps us to understand or be able to communicate. So there's no small contribution, and it's just being plugged in and helping out.
And I think that centers around this bigger picture idea of community engagement, because I do want to highlight that the amount of engagement we have from our community is astounding. And it also is remarkably noticed by stakeholders outside of the immediate patient and family community. And I want to make sure that everybody understands that the engagement and involvement that you have is noticed and is meaningful in everything that we do. It's, the center of our why. It's what motivates me. And I don't want to speak for everyone else, but I'm sure everyone else, what keeps us pushing day by day, your stories and your vulnerability and your trust are all so valuable. And we all thank you very much for that. And that level of trust and connection and engagement is so, it far surpasses the KIF1A.org patient and family community. It really is a key tenet in every aspect of activity that's ongoing within the organization.
Yeah, I mean, it sounds like soft stuff, but I mean, just your involvement as the community brought Dominique to do what she's doing, and what Dominique, you and Kat have done is unbelievable. I mean, once a month for 90 minutes, you've got 30 or 40 world-leading experts who come together to talk about a particular topic in KIF1A because of what you two do, and that's because of what the community does. It sounds soft, but it's really motivating. It's why people come to work and do what they do, and yeah,
it's very powerful. And definitely, again, we have to credit every single person, no matter what role they play in the community. And when I say community, I mean families, patients, researchers, scientists, doctors, donors. When I say the community, I mean this whole network of people, touched by KIF1A and who are joining our mission. So we appreciate you all for being here. And I think that's a good stopping point for this conversation, but I think these themes will continue throughout the conference. So thank you both, Dr. Dom and Hans, for kind of setting that stage for the rest of the conference, and thank you for your leadership and commitment to our organization. Thank you.
Thank you.